Research Frontier Reading Night. Do come forward, do take a seat. And we do have some incredible intentions. So first of all, it's a joy to be alive in a time where AI has made transformative progress. And DeepMind has this OG slogan. It says, solve intelligence and use it to solve everything else. And so in my mind, we're here for the everything else. That is, what are the applications of artificial intelligence to scientific research, to physics, to biology, to chemistry, to mathematics, which can unlock secrets, can unlock the understanding of the nature of reality that Elon's always talking about, can really bring us to a point where technological progress is driven by progress in artificial intelligence, as opposed to merely driven by progress in human intelligence. And so, in part, I want to create a context where we not only educate ourselves and write about AI for science, but actually come together to co-build, to mingle and hang out, to become friends, to really discover how to push science forward for artificial intelligence together. So today's the first big AI for science event. I want to thank everyone for coming. Thanks a lot. Good evening. Our intention's pretty clean. We're gonna basically teach one another through a learning experience. So basically the idea of the reading night is you have 2 hours of dedicated reading. And for a lot of people who are getting distracted, your phone's stealing your attention, it can be hard to set aside 2 hours of your life to read. But in a fun context where you can set aside those 2 hours surrounded by new people that you're meeting through a process of teaching one another everything that's in your papers, you get simultaneously a great learning experience, a great social experience, new friends, hopefully you enjoy it, and also hopefully you can build a community that's actually really impactful. So I'm really looking forward to it. The plan's pretty simple for the day. We're gonna have a quick talk from Andrew and myself, and then 2 hours of pin drop silence as we dive deep into the paper of interest. And feel free to pick a paper of your own choosing. I brought about 18 AI for science papers if you guys want to peruse them and check them out. But really, go into whatever you're most interested in that you can devote a solid 2 hours to fully comprehend and understand. And then we'll have group discussion at the end where we talk and discuss and debate and go back and forth over the details of these papers. And so, I guess I should say a bit of intro. So my name is Jeremy. Welcome to AGI House. In general, we've run a ton of really epic hackathons and events. We have a glorious view, which for me really inspired my writing. So when I was first here, I wrote 40 pages in 10 hours and felt incredibly intellectually productive. So hopefully you can hang out here or hang out downstairs on the floors below and have a similar profound intellectual experience. But with all that said, we're gonna have a quick talk on a literature review of AI agents in bio from the founder of Future House, Andrew. Let's hear it for Andrew.
Thank you. Awesome, this is so cool. Thank you, Jeremy, for inviting me, and thanks for coming out. I think a reading group is like the coolest hackathon idea. So I want to talk a little bit about a paper I wrote with Mike Cullis. So I'm co-founder of Future House. We're down a dog patch, and we're trying to do AI agents in science. We want to automate science. That's like the mission. I think a lot of people have this mission, but we're going to be we want to be tethered to actual experiments. So we have an actual wet lab. We have robotics with lab automation. We're doing real biology experiments, and we're trying to build this holy grail of AI for science, of a virtuous cycle of doing experiments and then responding to what you observe, and having an AI agent propose hypotheses based on the observations. So I'm going to talk about a review article I recently wrote. This is, I've been working on it for a while. We've been working on it for a while. It's not in its final form. It'll probably be finished in another month or so, but you can see it on archive. We have a version on archive. This version you can get. It's called a Review of Large Language Models and Autonomous Agents in Chemistry. We debated for a long time whether to cover chemistry and biology or all of science or something, and we came up with a pretty crappy compromise, which is that this review covers LLMs for chemistry and agents for all of science, because there's just there's not enough agents for science papers, so we could fit it on the review, but there's too many general LMs for a bunch of different areas. So one of the things I think is really fascinating about working with language models in science is we have to think about the data source. So we're familiar with language models in English, And basically what happens is that everything written down is something somebody thought was important or something that would meant to be communicated to other humans. So it's like a selective process in data that it's like either grammatically correct or it's somehow conveying an idea or somehow even reflecting physical reality. You go to biology and the thing which generates data is evolution. Like every sequence which we observe in nature is coming from some kind of natural selective pressure, so it's meant to accomplish something, or at least it's not detrimental. So you don't really see certain kinds of sequences in nature because they don't express into proteins or they don't work as DNA. So it's interesting, you can get like a similar process. But then as you get out of chemistry, as you get out of biology and you get out of sort of natural language, you do things like chemistry, material science, or physics, then in engineering, then the data generation process is actually really ill-informed. So like in chemistry, the biggest data sets are catalogs. Like people go through chemistry catalogs, see with all the compounds you can order, and they consider that the data generation process. It's like a big open question of like what it means to work with language models in these data. So I have this like kind of, what does it look like in chemistry? So in chemistry, like we look at these catalogs. And this is like the tokens, and this is if you want to train Llama 2, this is how many tokens you need. But there's like people also look at hypothetical compounds and look at things which can be possibly synthesized. But there is a massive source of tokens in chemistry, and that's the scientific papers in chemistry. But scientific papers in chemistry are locked away, unlike say computer science and machine learning. Chemistry is one of the most copyright intensive fields, so it's actually very hard to train models on this data. So I think it's a real open question, like how do you get around that? Do you have to so actually the current thinking for people at the forefront of AI and chemistry is to literally regenerate all of the data for the entire field for the past like 500 years with robotics, which is, so if anybody wants to take down Elsevier, it could save us some time, but I didn't say that. So like I said, I want to get you guys, we're trying to read here, so I'm just going to talk about a little bit of classification here. So in this article that we have, we break down all the different kinds of models by if they're encoder only, decoder only, and encoder decoder. So these are kind of categories of language models you're probably familiar with, and then they have different tasks. So things like captioning a molecule. So you guys are familiar with captioning an image. You can also capture a DNA sequence or caption a molecule. This is a category of things where you do multi-text. So this is it can be like a T5 or it can be an encoder decoder model. I won't get into these, but like I said, there's some of these encoder-only models that can do things like predict a property. You put in a molecule, get the properties of it, put in a molecule, understand what it is meant to do, if it's flammable, if it's safe, if it's a carcinogen, things like that. Some examples of these, there's Mulbert, it's a very famous paper. It's kind of an older paper now, maybe not as relevant to read, but I think it's a good starting point. And they trained it on guacamole. And so guacamole is this idea, okay, there's this hypothesis I don't think anyone's really executed, but in biology, a lot of the data comes from people doing bioprospecting or biomining. People ride around in a boat and drag, a little bucket out the back of the boat and collect samples, or they go around and collect dirt and then sequence it all because DNA sequencing is so cheap. You can just go around and look for new information to put in these models. In chemistry, we don't really have that, but guacamole is a bunch of natural products that as people try, it's a little bit harder to do, but you can grind up organisms into little pieces and then see what compounds fall out of them, and that's where this was trained. And then I just want to skip over this here, just talk through them. There's decoder-only models. These are the things like GPTs, these GPT models that basically you put in a chemical structure and then it can tell you about it. And I think one of the coolest ones, this paper, I think a lot of people just dismiss this paper because it was lampooned on Twitter because their PR campaign was a little bit too aggressive. But I think it's a really cool paper, is that they had this idea of building an omni model. But the modalities were not image, text, and video. The modalities were papers, code, reference material from tables, knowledge bases. Where's the encoders? Here it is, sorry. Text, LaTeX, delimited code, SMILES, amino acid sequences, and DNA sequences. They tried to build an encoder for all of these different things and build the ultimate science model. And I think it's a cool idea, and I think there might still be some direction to go there. But I think it's a great example of trying to build the foundation model for AI for science. Okay, so an example from Galactica is sulfuric acid reacts with sodium chloride and gives blank and blank, and then you have to fill in the chemical reaction as LaTeX. And it's interesting because it allows it to change encoders, because one of the problems with DNA, why language models are not good with DNA, is that they don't see them one nucleotide at a time. The word ACT is in DNA, and it'll say it as the word ACT, but it's also actually 3 nucleotides. And so these things can't even count DNA. So this is, I think, a really cool idea. But again, the results weren't great, but just they kind of killed themselves on it. So I hope Galactica a similar idea comes back again. Then T5s. Regression transformers, T5s, really underappreciated, I think, in the broader community, but T5s are really powerful ideas where you can put in like different kinds of masked data and then get out from another kind of data. So you can put in like some missing information and it can predict like if it's soluble, or some missing information, it can predict what's in there. But this idea of like having one model that can do multiple input-output tasks, really cool idea. Again, regression transformers for the first one, but I think BioT5 is like a great example of a model that tried to connect like drug discovery with genomics, with transcriptomics, and natural language all into one model. Okay, like I said, I'm just going to go through this quick. I don't want to spend a lot of time on it. So one of the famous models that's really like the backbone of many startup companies is the molecular transformer. This is a model which predicts, given the input chemical reaction, what the output products will be. And this like unlocked a ton of companies because chemical synthesis is one of the biggest like unsolved challenges, and the molecular transformer is just really average. And that was enough to, I think, get a lot of like Postera is, I think, a great example of a Y Combinator company that's a really big company, and all they do is do synthesis really well. Won't get into this too much, but then let's just take a second then and talk about agents. So what is an agent? So we're going to have a paper out of Future House soon about sort of a framework for describing agents and how to train them and try to like really characterize what is the problem to be solved from a mathematical perspective and reinforcement learning. So trying to formulate sort of this Markov decision process, so MDPs as the background for agents. I won't define it today. We'll have a paper coming out. You guys can read all about it. But basically, an agent is a policy in a route. It looks at an environment, looks at observations, and then makes a decision that leads to some reward. So we have in this article, like our kind of nomenclature of a language model agent, but I think this is like the proto-architecture. Like I said, we're coming up with something more mathematical. One of the things that bugs the shit out of me for AI for science papers with agents is that it's all cartoon-based. It's like code, prompts, and cartoons. And we're trying to get to something more grounded. So I'm really looking forward to that. And James here is one of the top engineers on the team working on that. So like I don't know if it's the original, but I think one of the most famous scientific agents papers is the ChemCrow paper that was done by me and Sam Cox. And then we work with a group at IBM that did the robotics. And this is basically trying to do the whole thing of connecting automated laboratory robotics with natural language tasks. So you can go from like make an insecticide, and then you have like the model trying to find out what are common insecticides, what is their structure, plan a synthesis route, go to the robot arm, it makes the actual compound, and then we check to see if it comes out. And I think one of the things that we were most proud of in this paper was where we gave it the supporting information from a paper which had a bunch of data on color and molecular structure, and we said, make a new molecule with a specific color. And it built a machine learning model with Python code, like found molecules that had a specific color, went and then made the actual color, and we like took it off the the HPLC column, and it actually had the color that we wanted. Well, within 15 nanometers, but that's pretty close. Awesome. So I got a faster here. So paper QA. This is another upcoming work from Future House. This is a superhuman literature research agent, and yeah, great. So I don't want to talk about anything else, but I will say one thing for tonight that maybe is useful is we have this website, hasanyone.com, and you can go here and you can say like, has anyone tried to prove the Riemann hypothesis via crystallography? And it will launch a little scientific agent that will go research and read papers and tell you, no one's tried to prove the Riemann hypothesis via crystallography. And then it tells you about the papers it read to come to the conclusion, and you can use this as a way to see, has anybody done something before? So when you guys are thinking about new hypotheses, try that.
Does it have an API?
Yes. So I don't have any details on it yet, but we plan to do a hackathon later this month. It's August, in September. We will do kind of a hackathon with access to this kind of an API, and we want to see what people can build with it. So we're really excited to see what people can do. But this one, just if you go and check, has anybody done something? Check it out. So that's a whirlwind. Like I said, you can read the paper. It's called A Review of Large Language Models and Autonomous Agents in Chemistry. If you see any missing references, please let me know. It has a ton of bibliometrics, like big, like phylogenetic tree of research papers, like which papers evolved to which method, which categories, like counts. We actually built an agent which reads through papers on an archive email list every week and opens them up as GitHub issues to add it to the review paper. And then we say, oh, this is a good fit. No, this is not a good fit. So we've been working on this for a while. There's a GitHub repo with all the papers, and I think it's a cool paper. But, you know, it's not necessarily a deep read, so you guys can read something else. Thank you very much. Wait, questions for me? Oh yeah, we have job openings, so if anybody wants to do this for a living, you know, come up.
Quick question.
Yeah.
In terms of the problem with getting access to all the chemistry papers,
Yeah.
so chemistry not my field,
Yeah,
but the open access push,
yeah.
I would imagine would be a solution if everything's or is chemistry just
No,
carved out?
you are absolutely right. There's a problem with open access though, and there's two problems. One problem is that the copyright doesn't always like go into the public domain. So there's actually an issue where like open access doesn't just equal that the paper can be used for anything. There's things like, you know, like for example, CC BY ND. I don't know, we haven't solved. Does CC BY ND prevent model training on CC BY ND work? I don't know actually, because is that derivative? Or if I answer a question with a CC BY ND paper. So I think there's actually some issues, but in general, yes, open access papers make up a big portion of that, but it doesn't quite solve the challenge yet. So yeah, it's definitely stuff. Yeah.
What do you think about the state of citations? Are they accurate out there?
Oh my God, there was a hackathon. I think a few months ago, I was here for a hackathon, and somebody built like what was it called, like unfaithful citation bot. It would read through papers and look at the citation and then see if the paper could plausibly be connected to the sentence. And you know, obviously they weren't. But like one of the biggest problems we have with like hasanybodyDone.com, hasanyonedone.com, fuck, I can never keep it straight, is that like open access is like inversely correlated with journal quality a lot of the time. So papers which are easy to get are actually not good quality papers. So we had to build like a big database of quality of papers and if papers are retracted and use that to assess whether we should include an article or not. And you know, it's not exactly the same issue what you're describing, but definitely like quality of papers is something which is approaching an urgent need, especially with Gen AI tools leading to this. Like I think there's a journal, what was it, like Deepy, what is his name on Twitter? He was like saying there's this journal, it's like $50 to get a paper published and you can use it to build your E1B visa application.
Yeah. Pay to play.
Yeah, and so people just go in
What's the name
ChatGPT.
again?
Yeah, no, I'm not gonna say the name. But basically, this is like
Can
an
I
issue.
also buy citations?
Yeah. And in fact, one of the hardest parts is we use LLMs, and the LLMs are actually really susceptible to strong claims. Like I came across a paper put up on Twitter about like this paper saying if you eat 12 Oreos a day, it's better than taking statins for heart disease. And this came out of this LLM, and I was like, what the fuck? Like, that's not real. And I look out, and somebody put this crappy paper of like they followed one dude for 16 weeks, and 16 days, and they published that paper. N equals 1, 16 days. Okay, I'm way amped up on
How did you address that?
Huh?
How did you address that?
How did I address that? I couldn't, because that journal was before a good journal, but has been shitified. And so now I don't know how to solve it. The one clue is like the time from submission to acceptance was 16 days, which is coincidentally the time that the man was eating Oreos, or woman, I don't know. So I think time from acceptance to publication is how I view that signal. We haven't figured out how to put it into like an LLM flow, but yeah.
But you already tweeted about it. It's like some way of confirmation.
Oh,
Yeah, like validating the research.
yeah, that's right. If you put it authoritatively as a claim on Twitter,
Yeah.
and if people get pissed, then that's a good signal that it's not real.
But citations doesn't work? Like how many times the paper has been cited?
We do use that as a signal that there are citation farms. You can cluster them out, but there are citation farms where people just cite each other's. Like one of the guys with the most citations in the world, what he does is every day he wakes up and he writes 3 letters to editors, and in these letters to editors he cites himself 16 times and makes such a like nebulous claim that like a ChatGPT claim that more research is required, that the editors let it fly and get published, and this guy has like, you know, he gets 500 citations a week from doing this process.
Wow.
Well, that is such a cool outfit.
Legendary.
Yeah, all right. Happy to chat about
Cool.
this.
Thank you. Okay, so I'm going to walk us through like intros to everyone, but before we do, I want to say I run a company called Omniscience, and we do paper generation. So if you've done a lot of great research work and you describe all that work to the paper generator, it'll write a draft for you, which can save you a ton of time. I can also do literature review. So I wanted to actually ask if anyone has a scientific paper they've always wanted to read that perhaps does not exist. and we will conjure it into existence, and perhaps you can read it. Andrew.
Okay, yeah, there's this like connection between the Riemann Zeta hypothesis and one-dimensional quasicrystals. So I want to see like, I want to read the paper that explains this connection, and you know, if I can go out and do crystallography to solve the Riemann Zeta hypothesis.
Has anyone done that?
We'll find out.
Connection between the Riemann Zeta hypothesis
And one-dimensional quasicrystals.
I see, one-dimensional, essential. Okay, great.
Quasi, not
Quasi.
quantum.
Let me see. Quasi. Okay. And then,
Now I'm trying to think what a
is
quantum
that
crystal
correct?
is.
Okay,
Cool.
wonderful. So, if you want to try it out, go to omniscience.tech/demo, and you can make your own for the event. But let's do some intros. So you guys are all wonderful people. I basically want to do a lightning intro. So everyone ask a question, which should end with a question mark, that you're curious about the answer to in AI for science. Could be in any domain, could be in physics and bio and math, and it could be a meta question. But just tell everyone your name and one thing you're curious about, so that when we hang out, people can come up and say, oh, by the way, I have the answer to the question you've always been looking for. So I'll start real quick. So my name's Jeremy, and yeah, I guess the question that I always have is about like representation. So like, what is the abstract representation of knowledge from which scientific research is coming, and how can we mimic that representation?
That's a great question. It's a great question.
Gosh. Yeah, I guess scientific knowledge is good. How can we replicate it? You have to get
Yeah, I'm Mitchell Reynolds, and I mainly I don't really have a question. It's more just like reviewing the current landscape of the AI tooling
Okay, for scientific research or generally speaking?
for scientific research
Okay.
and PASTA.
Pasta.
That's an acronym for Process of Automating Scientific and Technological Advancement. It's a really nice acronym.
And delicious.
Yeah,
Did you just come with that, or is that a thing?
it's a thing.
Oh wow. Okay. Wait.
I'm Alex Kogan. I want to know what happens to PhD students with a K.
Oh, sorry.
Yeah.
Just generally.
Yeah, like if like science gets automated, like do you
Oh,
still need
I see.
PhD students? And yeah, what happens?
Let's like go on this side of the room first.
Victoria, Wiki, probably, but it's really fine. Victoria Lex, yeah. WKS. So I would, is, curious for me is how do you combine and mix and match models on top of one another to kind of produce like a multi-omics, ideally, adaptable model that can answer things holistically, but technically.
My name's Luke. Short here. CHTIU. And my question is like how we can create models faster on with less compute.
So go behind you.
I know, and then you can say, I just want to know how often just not make mistakes.
How hallucination?
Just how often does it make mistakes on every.
Oh, okay. How
How
often?
minimize when it's like making mistakes. Either wrong data or
Hi, I'm Sandro.
Cool.
I'm interested in AI generated models to predict how to build 3D structures.
Oh, okay. Cool. And I guess for chemistry, for
For biomedical
bio,
professions.
for biomedical. Cool, okay. Yeah. There's no- Or if you want to go, yeah.
Rob, and I'm just learning last name is Barcelona. So,
Cool.
I guess I'm kind of curious about data composition for large language models and how much more efficient you can get at scheduling with data curation for data composition.
people have been adapting it, and in meta they do a lot of complex stuff. Would you answer that?
So Yannick, D-Y-A-N-N-I-C-A, D. What are some efficient methods to improve the sample efficiency of molecular generative models?
Like AlphaFold?
No, it's a small molecule.
Oh, small molecules. Oh, cool.
Small molecule.
So not. Nice.
I'm Elizabeth. My last name is Trykin. And I think my question is around how do we generate more data in biology to actually fuel the models that are being built.
Yeah, they're very data hungry.
My name is Daniel Bransov.
Yeah.
And question, is there any examples how people use computable ontologies like basic formal ontology or derivatives with LLMs? And how it help agents to reason?
A lot of thoughts. Chat.
I'm Ash. I was looking into some of those papers, and there's a common theme around Occam's razor. You know, like, let's minimize the number of parameters so the model is more explainable and it makes a lot of sense for science. But the question that I'm curious about is, what if the trade-off that they should look for is not the number of parameters, but more like a different architecture, like Kolmogorov-Arnold networks? So like, if you have nonlinearity on every element, you probably need even fewer. So maybe like some of the state of the arts can be just replaced, switching to like to this architecture, and there's more than enough training data because most of the stuff coming from papers is in simulations.
The entire bias variance trade-off was like thrown into dust when neural nets came along, and double descent makes it clear that we have no idea what's going on. So it's totally unclear that this is a measure of complexity that anyone should pay any attention to. But yeah, every time I write about the Occam transfer stuff, I'm just deeply skeptical and pray that they find Jesus.
I'm Aditya Advani, and my question is, if you could burn 500,000 tokens a second, what kind of agent framework for science would you build, and what would it be able to do?
Cool.
I mean, I know what I would do.
I'd love to hear it.
Yeah, it's okay.
Andrew White. And I think the biggest question, what I work on every day, is how do you make scientific agents train? How do you make them learn?
Very big question.
I'm Miles.
Yep.
I'm interested in how to use tools from sequential decision making to do efficient synthesis planning.
That's good. And the synthesis here is between like distinct stages in an agent pipeline, or distinct ideas being merged in a compositional way, or some other form of synthesis.
In this case, it's very specific to chemistry.
Okay, I see. So like specific chemical molecules being combined with each other.
Yeah.
Cool.
TJ Burnett. I'm gonna riff off yours. So you had you've scrolled down, but so are there different levels of abstract representation of knowledge that each scientific problem uses, and how can you differentiate between the different levels that are necessary? I don't know how to.
Yeah.
Can you coarse grain knowledge?
Can you coarse grain it at different levels and different contexts?
Yeah.
For science,
Yeah, I built a bunch of systems that construct the hierarchy and use various four levels for various things. Maybe we should chat about it. Yeah.
I'm Vahe Barakanyan, so Vahe G is good enough. So I'm mostly interested in generating models for material science and how to use inherent symmetries in materials to construct better materials.
To construct, so it's a generative model for novel materials? Okay. So there's like some structure in the data that, there's some architectures of just money, but they could take advantage of. Cool.
So I'm Casper, it's K-A-C-P-E-R. And I want to get up to speed with what's kind of the state of the art in terms of post-training, specifically for things like chemistry, maths, or coaching.
Are you in ChemNLP?
No.
Okay, there's a Discord called ChemNLP working on this exact problem. You should join it.
Alright. Yeah, I actually started with in chemistry before. So.
Oh, okay.
Yeah, I mean, it's accessible too. It's like cheap to do this post-training, relatively speaking, so lots of room for innovation. And we'll probably try to hit the math and chemistry hackathons, do custom post-training, so should come build some stuff. Behind?
Jason. I'm interested in the nature of effective hypotheses versus just novel hypotheses, particularly in discovery and biological sciences.
Yeah, that's very big and complicated prior. But yeah.
My name is James Hill. I guess my biggest question would be how can you systematically search the hypothesis space given the data that you have in a scientific context? But it's kind of general, but it's relevant.
Yeah, very similar question.
Similar question.
Yeah, hypothesis search. Rob?
Yeah, Rohan. How much is science constrained by intelligence versus experimentation?
Oh, that's a great question.
Yeah, like definitely math is intelligence constrained, but I'm not sure science is.
I think about that a lot. Like, what's the upper limit, of intelligence for helping in science?
Yeah, this question is always like poorly formed too, because the measure of intelligence that you use is relevant, and it's unclear at what scale, like how much more intelligent exactly, like how many more experiments, and like how much the type of experiments differ. But
like how many PhD students does it take?
Yeah, there's a decision here, really. Like, if you're a PhD, should you like on the margin, like be an experiment generalizer versus intelligence improver?
Exactly.
It's really Justin.
Yeah, Justin, I guess the biggest question is what will the infrastructure look like for hosting the models that are agents that we use for science?
It's like compute infrastructure.
Yeah.
Like, is it massively parallel? Like, do you need tons of compute? Kind of questions.
Denisa?
Yeah.
My question, I guess the biggest question I've had is whether or not AI can help us understand objective reality, or whether or not it be similarly limited by us humans and how we view things in the course of the world.
Yeah, like imposing the language as an initial representation does feel like it destroys a lot of the potential magic. So it's an important question. Yeah. And last but not least.
My name is Shankar.
Yeah.
And my question is, I think, the same lines what Andrew Wade said. So when you are building agents that are basically done for domain specific, how do you make them like unlearn what they have learned so that we can make them perform better for domain specific tasks? So I'm not sure any of these papers will be better, but I'm curious to learn more about that.
So I guess the intuition here is that usually it's by violating some pre-existing assumption that you make scientific progress. And if the models are going to take for granted everything you already know, it's going to be hard to make the kind of progress that came from having made a mistake in your initial representation of the problem. Something like that. Cool. Okay, well, that's a fascinating overview of everyone in the room. So hopefully you all found someone to talk to.
Oh, I have one more question.
For sure.
How much of science is continuous versus discontinuous? That's kind of along the same point.
Ash, you might have something to say about the continuity thing. We're gonna do like a discrete models only hackathon.
Yeah, definitely.
I mean, I think the reality is quantized, so there's definitely some limit.
Planck length. Yeah, fascinating. But yeah, continuous approximations are so high quality. It's kind of crazy. Kind of scary, don't you think? Maybe should think hard about it. Okay, that's very cool. Yeah, I just got a sense for who's in the room, what they're up to, which is very nice. So very high level. This is going to be a part of an AI for Science series. In mathematics, there's been a number of breakthroughs. So DeepMind came out with their performance of an alpha proof system. There's no paper on it, but basically showed that at IMO they were able to get a second and solve a number of very hard IMO problems. And all these systems build on the Lean system. I don't know if, who's used Lean before? You know that? Okay, small handful of people. So basically, it's a language that allows you to write mathematical proofs and come to conclusions about them, and they train models to work in Lean and manage to do a really great job with IMO proofs. Quen2, so this is like a Chinese model. They actually have state-of-the-art and open source and are outperforming alpha geometry in important ways. There's also no math-specific version of the Quen2 paper, but yeah, we intend to basically have a hackathon with models like this to try to do novel mathematics, to train models, to fine-tune models and post-training, and also automate discovery. In my opinion, automating machine learning research and automating mathematical research should converge, but the math research that matters is applied math research, and so systems like Wolfram Alpha that let you generate infinite amounts of, or as much as you want of applied math data are very interesting. Yeah, in physics, there's an org called Polymathic AI, which has been putting out fantastic foundation models for problems in astrophysics, and certainly has been attempting to create high-quality simulators. So they have this sort of multiple physics, pre-training setup, which, I guess much like the paper Andrew was presenting around trying to build a comprehensive foundation model with many different input types that go into the training process. Attempts to do that in physics. There's been less overt progress as there's been in mathematics with loud announcements, and so I really hope that physics can catch up in some sense. In bio, though, there is a ton of action, including ChemGrow, and action to come. AlphaFold has already been a huge deal. So actually, Demis became CEO of Isomorphic Labs, which is a company DeepMind spun out specifically because of AlphaFold. And it is just hard proof that if you work hard, you can do incredible work in biology with machine intelligence. The future of genetic editing looks like it might involve novel variants of CRISPR that are invented by machine learning algorithms, like OpenCRISPR-1. And the ARC Institute has also been putting out their own foundation models. So we'll actually get them in for an event too, which will be great. And finally, I think there's a big opportunity in the inverse problem. So with AlphaFold, you're generating the protein, but in a lot of cases, you're trying to latch on to a particular site. And so the question is, can you give that site to a model and have it modify or construct a molecule that will attach effectively? And so models like DiffDoc are in the direction of this question of, how do you basically work on this problem, which is essential to things like drug discovery, where you want to modify a molecule in a way that leads to it binding appropriately to some protein or capsid of interest. Yeah, I guess brain computer interfaces. So we had a huge BCI hackathon a few months ago, and we're gonna have another. But in my mind, the intersection of AI and brain computer interfaces is deeply profound. So actually, can change the way that we think, the way that we communicate. Some of these papers are about basically neuroprosthesis. So if you can't speak, you can have a model with access to your brain signals speak for you. Similar story for writing via text. There's a company called Synchron that 2 years ago had people tweet from BCI. Neuralink just put their BCI on their second patient, and they're playing video games. A lot of neuroscience, in my mind, is bottlenecked by hardware quality. So this is a very big deal. And then I guess there are a few overviews of science, generally speaking. So if you guys are interested in those papers, I have all of the papers that are listed here accessible. So 1% grab all of them. And then finally, there are a ton of AI tools for research. These are 8 of them, including PaperGen. And yeah, I guess if you are creating an AI tool for research and you'd like to pitch it or present it, definitely come talk to me. We'd love to show them off. And I guess that's perfect context for showing off Andrew's paper, which is ready, which is on the connection, exploring the interplay between the Riemann Zeta hypothesis and one-dimensional quasicrystals. And so, you know, it opens with an introduction, a description of the Riemann Zeta function. I don't know what you're looking for, so if you want to control this right now.
Oh, I have no idea.
I haven't seen these papers. So.
I mean, this is like one person said this in like, I think a letter, you know, and nobody's really looked into it. So this is the frontier of knowledge.
Oh, cool. Okay. So here's the original hypothesis around how all non-trivial zeros, the Riemann Zeta function, have a real part equal to 1/2. And yeah, it does an overview of the sort of fundamentals of quasicrystals.
So quasicrystals have to be described with complex numbers. And I think they're like zeros to a function actually. I'm not sure, but like this is the idea: is that the Riemann Zeta function is about one-dimensional like roots and complex plane, and so there's supposed to be some kind of connection.
I see complex number work.
Yeah,
interesting. So yeah, it covers the Riemann Zeta hypothesis in mathematics, and then does a sort of physical description of phazon flips and quasicrystal stability.
This is so much better than Sakai AI.
It's true. Yeah, no, it like dives into deeds on quasicrystal growth and formation.
That's good.
And that merges them. So analytical techniques for zeta functions and quasicrystal structures. So here's the zeta function regularization and quasicrystal energy levels. Spectral properties of quasicrystals can be related to the zeros of the Riemann zeta function, as suggested by the trace formula, which connects the spectrum of an operator to the geometry of the underlying space.
Good. And has anybody done.com says this is a novel result, so.
Oh, you checked already? Oh, no way. That's hilarious. Like, how'd you get the paper?
No, I mean, like, this question. That
Oh, I see. There's no. You looked for it.
I looked for this question and didn't find anything. So this is it.
Yeah. So, yeah, I mean, we should certainly get a mathematician to take a look, but. Yeah, diffraction patterns and the distribution of primes.
You guys could be looking at a Nobel Prize right here.
Yeah, I mean, this is pretty crazy. Spectral properties of quasicrystals. And a relation to the zeta function, so the connection arises from the observation that both systems can be described as similar mathematical structures.
Cool.
So refocusing on the trace formula, which relates to the spectrum of the Hamiltonian to the geometric properties of the system, which has a counterpart in the explicit formulas of number theory that connect the zeros of the zeta functions to the distribution of primes. Yeah, I will send you this result and let me know if your friend is satisfied by this mathematical.
The king of Sweden.
Yeah, I give you Omniscience. If you would like to read like this or other papers, go to omniscience.tech/demo. So I made it free for you guys, and you can just describe your paper, and you can read whatever paper you want to do. So feel free to take advantage.
It hits login, by the way.
Yeah, there's a login. That's true. You do have to log in. It will email you the paper. It takes less than 10 minutes, but it still takes about that long. So yeah, it's not immediate. I'm sorry. We'll get there.
500,000 tokens per second. That's all you need. Right?
We'll get there for sure. Okay. So I guess great. So at 7:30, we're gonna transition to 2 hours of paper reading. Oh, so you have a question?
Yeah. So do we do 2 hours of paper reading and then present the papers that we're reading, or how does that?
Yeah. So so we're gonna probably have like 2 big group discussions, and you'll have a conversation. You'll describe your paper to the group, and then have a conversation about it. We'll spend about an hour discussing everyone's papers. Maybe 3. The numbers are a bit much, but yeah, something like that. And the intent is to try to finish your paper. You can read multiple papers if you want, or do a write-up. There's a version of this event where everyone writes a review or like a reaction to the paper that they read, and then that write-up is put online publicly, and we co-launch our reviews on Twitter, making a public conversation about the papers in question. And you know, if you have enough people who are expert enough to comment on your review, that can be a like awesome public discussion about some interesting part of science research, and then people on say like Twitter or wherever you've been discussing it can come in with their own commentary, or you can mention the authors or something like it. But I was gonna do that tonight. I was just gonna have a chat. So let me know if you have any ambitious ideas for like how we could do something like that though. It'd be cool. Other questions? We good? Okay. So yeah, here's how it's going to run. Basically, it's 7:30, so we're going to read until 9:30. This is a like 4-story mansion, so do not go upstairs. That's where private rooms are. Feel free to spread out on this floor, the floor below it, and the floor below that. All those floors are open for paper reading. So find a quiet corner, settle in. My request is that you feel free to talk for the next 5 minutes, and then at 7:40, I'm going to cut talking, and it's all silent, focused reading they're talking about. And then we'll regather at 9:30 to discuss everything that we've learned. Any questions?
Do you want people to split up by papers within topics? Like I would, like if other people are reading AI math papers, come to me and we can like distribute papers.
Yeah, so if you want to hang out with math folk, that's cool. You should try to read and like primarily like complete the readings that way. If you think there's a way you can talk and not distract other people because you found a room or a corner, you know that that's a better use of your paper reading time than paper reading, then I trust you to violate the norm. But please be very judicious about that and don't abuse it.
Make sure I'm saying in the next 5 minutes.
Yeah.
Like
Next 5 minutes, do whatever you want.
Amongst the AI math people, we can like choose 3 papers and split it up amongst
I like that.
ourselves.
Oh, for sure, yeah. No, the strategic, like, collective reading agenda is a great idea. Yeah, 100%.
So if anyone is reading AI math papers, feel free to come here.
Yeah. And if anyone else wants to strategize about splitting up reading duties, that also is great. So feel free. Any last words?
You want AI and bio?
Okay.
I'll go for bio.
Yeah. 3, 2, 1, read.
You want to go?
Let's do that second one. That's coming from experience.
Yeah.
There's a bunch that I was looking for. Like obviously there's like the big reading papers like which one are you reading?
I don't think they like official overview paper, research paper by like Joshua Pages. Oh, how does it feel? And then someone said pasta, and I'm definitely gonna look that up and figure out what's going on there.
If I knew this changes the game and cursor on Twitter, it actually is like
yeah, let's see, right?
Generally I'm just like